The University of KwaZulu-Natal College of Agriculture, Engineering and Science is committed to making a difference in our world, from the classroom and laboratory to the field. It provides one of the finest study and research environments on the African continent and is recognized internationally as a center of excellence. Agriculture, Engineering and Science is one of 4 colleges that make up the University of KwaZulu-Natal and is home to over 8,700 students and approximately 700 staff. 3 of the university's 5 campuses form the core of agriculture, engineering and science activities in the region. The college comprises 5 schools: agricultural, earth and environmental sciences, engineering, chemistry and physics, life sciences and mathematics, statistics and computer science. Engineering is offered on the Howard College campus, agriculture on the Peter Maritzburg, and all the other sciences on both the Peter Maritzburg and Westville campuses. Exceptions are agricultural engineering, which is offered in Peter Maritzburg, and geology, which is offered at Westville only. To compete with the best and to meet the challenges of the 21st century, South Africa needs highly skilled professionals in the areas of agriculture, engineering, and science. We offer a wide array of interesting and innovative degree programs that open doors to a variety of different careers. Some degrees are unique to UKZN and are not offered at other tertiary institutions. For example, we are the only university in South Africa to offer a fully accredited agricultural engineering degree, and one of only 2 universities in the country to offer a BSc in land survey. Our postgraduate food security training program is the only one in the world, and we have the widest range of agricultural disciplines at any single South African or African campus. The choices of degree and the options on offer may seem confusing at first. Engineering degrees are more specific, and the options are usually career-focused. Emphasis is placed on training in the specific principles which engineers deal with in their professions. Engineering offers 7 different professional specializations. There are also degrees in land surveying or geomatics and property development. In science, there are structured or career-focused degrees. These include fields as varied as chemical technology, marine biology, industrial and applied biotechnology, and computer science and information technology, to mention a few. There are also more general degrees with a lot of flexibility. In this case, students have the choice of studying a BSc M stream in maths, physics, computer science, and statistics. All of these have a strong mathematical component. The other option is a BSc LES stream with a focus on the life and environmental sciences with subjects such as geography, genetics, botany, hydrology, zoology, ecology, and microbiology. In agriculture, there are various different options that involve practical and scientific applications. Students can combine science subjects with specific agricultural disciplines for a 4-year Bachelor of Science in Agriculture degree. These are more career-orientated. Agriculture and agricultural management are 3-year degrees with a higher practical component. Dietetics is a professional qualification that deals with the role nutrition plays in the promotion of health and the treatment of disease. Most degree programs across the college have field trips and projects that contribute significantly to the curriculum. This ensures that students learn in real-life situations under full field conditions. Many of our lecturers have received Excellence in Teaching awards, are recognized internationally, and are rich in practical experience. Students have access to excellent facilities that rank amongst the best in the world. 300 million rands has recently been invested in new teaching and research facilities, state-of-the-art laboratories, and improved equipment. The 30 million rands new School of Engineering building is the end of engineering campuses across the country. Agricultural students have access to a 356-hectare research and training farm situated near the Pietermaritzburg campus. This facility provides students with hands-on training and is used extensively as an outdoor laboratory for teaching and research in areas such as animal and poultry science, horticultural science, and agricultural engineering. Adjoining Upalinga is the Bisley Valley Nature Reserve, which supports a diversity of plant and bird species, as well as some large mammals. It provides a rich learning and research environment for staff and students. Based on the Westville campus, the college possesses unique facilities for research into higher voltage power transmission. These facilities are available in only 3 other places in the world and form part of the ESCON-sponsored Science and Technology Innovation Park. The Science and Technology Education Center showcases the college's scientific achievements and promotes the public understanding of science, engineering, and technology within the region. The college houses a wide range of sophisticated equipment
Good evening, students, and welcome to our Matric Helpline session, the second one for this period. And we are going to be doing genetics tonight. I'll just ask Ntokozo to please make me co-host so that I can share my screen. Okay, thank you. Just give me a moment. I'll share my screen with you guys. And Chokoza, you can continue playing the video. I'm not sure why it stopped prematurely. I'm sorry, my PowerPoint is just taking a while to open.
Its microscopy and microanalysis units, staffed by highly qualified and skilled technical specialists. The College of Agriculture, Engineering and Science prides itself on its graduates who are highly sought after. In a market characterized by a shortage of qualified professionals in science and technology, our graduates are readily employed around the world in the fields of agriculture, science, commerce, industry, engineering, and research, to name just a few. Research plays a major role within the college and operates at the cutting edge of technology. College has the highest research output in university and produces the most research publications. There are over 60 well-established research groups and centers within the college. The African Center for Crop Improvement caters for students from all over Africa. It is one of only 2 African plant breeding programs on the continent. Based in the School of Agricultural, Earth and Environmental Sciences, the African Center for Food Security focuses on alleviating hunger in Africa. It offers food security training programs, undertakes academic and applied research, and provides policy and program support. Promoting sustainable engineering practice is the Center for Research in Environmental, Coastal, and Hydrological Engineering, or CRESH. It deals with the interaction between engineering and the natural environment. The Mechatronics and Robotics Research Group in the School of Engineering provides an excellent research and education environment for its senior undergraduate and postgraduate students. Judged one of the best research centers at the university, the Center for Plant Growth and Development focuses on high-quality research in the plant-based disciplines of plant physiology, plant molecular biology, and ethnobotany. The Center for Quantum Technology deals with physical phenomena at the microscopic level. Quantum physics led to the development of the transistor and the laser, and is currently being used to develop quantum computers and quantum cryptography. The college is committed to access
Okay, students, I think Ntokoza has lost connectivity. It's okay. We can proceed with our session for tonight. It's, as I said, on genetics. Okay. I'm sorry if I sound terrible. I'm just a little bit under the weather. So I'm going to start screen sharing. Okay. Can everybody see my screen? You can just feel free to give me just a thumbs up, or you can just unmute and say yes. No problem. We are very
Yes, we can see your screen, Jerusha.
okay. Thank you.
Yes, we can.
Okay, great. Okay, guys, so don't feel shy. You can unmute. And remember, if for those of you who are new, who did not come last week, Friday, this is a session where we going to revise the section of genetics. You are more than welcome to stop me at any time and ask questions or give an answer or anything relating to what the work we are doing. You are more than welcome to unmute and say what you need to say and also ask questions. Please feel free, and this is for you guys to help you, so please do not feel shy. If you are really feeling shy and you don't want to unmute and speak, you are more than welcome to put your question in the chat, and my colleague Yanga will let me know, and I will be happy to answer your questions. Okay, and I will be happy to address any concerns you may have. Okay, so please feel free tonight, and it's very chilled, so just give me the answers as we go along, and I hope that the session will be helpful to you. Now, the best way to do well in genetics, which is actually the second biggest section for your matric year apart from evolution, is to practice. Okay, so if you the more you practice your questions, the better you're going to do at it. Okay, so you will see that in genetics there's a few patterns that pop up in terms of how they ask the questions, what they expect from you in the questions, and also yeah, the types of things that you're going to be asked in genetics. You'll see there's common questions that keep coming up. So you should take note of these and make sure that you are able to answer them. Okay, all the questions that I'm going to be doing tonight are from past year papers, so it is good practice. Whatever past year papers you can get your hands on, you must do them. That is the best way to prepare yourself for the exams that are coming up. Okay, your June paper, I'm sure you know, is just one paper, so we're gonna be covering all the topics that's going to be in your June paper. Okay, so I'm going to be doing one topic per week basically, but please feel free to post any suggestions in the chat. Okay, my colleagues have also posted links in the chat for you to register, so please may you kindly take a second and just click on that link to register yourself onto our system, where you'll be notified of further Matric Helpline sessions that we will do later in the year. Okay, and we'll really appreciate if you could just take a second quickly to do that. And then at the end of our session tonight, you can also click on the link in the chat that allows you to evaluate and provide feedback for our session. Okay, so we would really appreciate if you can take a second to fill in those two quick questionnaires. It's very quick. The one for registration, just your email and your name and probably what school you're from. So please fill that out if you can now, okay, while we are still giving students some time to log on. Okay, so I'm going to jump straight into it. We're going to start off with some genetics questions. We'll start off pretty easy and then it gets a bit harder. So for the genetics section, you definitely will get some of these in the multiple choice section. Okay, so if you get a multiple choice genetics question, it's probably going to be a loaded question. They can even ask you to do a Punnett square for your, the question can require you to do a Punnett square, even though it's multiple choice. So don't take it for granted that if it's in a multiple choice, it's going to be easy genetics questions. The hardest ones are the multiple choice genetics questions. So Therefore, it'll be easier for you guys to actually learn off some of the patterns that you see in your offspring. Okay, for your F1 generation, you need to know that when you cross a monohybrid, in a monohybrid cross, sorry, when you cross a homozygous dominant with a homozygous recessive, all your offspring are going to be heterozygous dominant in terms of the genotype, and they're going to show the dominant trait in terms of their phenotype. Okay, so you need to know patterns like that. What will the F1 generation be when you cross two heterozygous dominant individuals? What will your genotype be? What will your phenotype be? Okay, so you can make those notes for yourself. I suggest you do that so that it will help you when you're studying. So then you know immediately when you read a question that, okay, it'll be a multiple choice question. And if I see a heterozygous dominant crossed with another heterozygous dominant, immediately, what's my genotype? What's my phenotype? Because that's what the question will ask. Okay, so you can answer those questions much faster if you actually write down what your genotype and phenotype is for, let's say, two heterozygous dominant parents, one heterozygous, one homozygous, and two, so two heterozygous dominant parents and one heterozygous, one homozygous dominant parent. Okay, so write down your genotype and phenotype that you're going to get from those parents and learn that off. Okay, I hope everybody's with me. If you don't understand, please just shout. I'll explain myself again. I'm sorry that I sound so bad, really. I'm sure I sound terrible, but it's just this, I have a bit of a flu, so my voice is not cooperating. Nevertheless, I hope you do learn something tonight. Okay, so I'm going to start with our first question. Please unmute and feel free to answer. I'm going to type up the answers for you here as well, if that's more helpful to you if you need. Okay, maybe some of the long answers I'll type up. Okay, but you can take note of these answers as well. You can just jot them down as well. Okay, so in a plant species, two characteristics, flower color and plant height. Okay, so now immediately straight off the bat, sorry, I didn't start off with the easy one actually. Two characteristics, so that immediately tells us dihybrid cross, flower color, plant height were studied. Each of these characteristics has two variations. Flowers may be red or white, and plants may be tall or short. Plants that are heterozygous for flower color have red flowers. Plants that are homozygous recessive for plant height are short. So what does that immediately tell us? The dominant trait for flower color is red. The dominant trait for plant height is tall. Okay. So they have given you the letters to use there. Okay. So what is the term given for a genetic cross involving two characteristics? Answer, anybody?
A dihybrid cross.
Very good. Okay, you already won yourself 1 mark. Well done. Okay, next, give the dominant phenotype for flower color. So what's our
Red.
dominant type? It is red. Very good. And how do we know this? They can ask you that as a follow-up question. How do we know this? The F1 generation are? They are red because it says plants that are heterozygous for flower color have red flowers. Okay, so that tells us if you've got a dominant allele and a recessive allele, the color comes out red, which means that red has to be dominant. Recessive phenotype for plant height. What's the recessive phenotype? It will be homozygous recessive for plant height is
Short,
short.
It'll be short.
Okay, good. Phenotype for a plant that is heterozygous for flower color and homozygous dominant for plant height. What's the phenotype going to be?
Will it be tall and white?
A plant that is heterozygous for flower color. No, read that part again. See, it says heterozygous for flower color have red flowers. So heterozygous for flower color, red.
Oh, right. Okay, sorry.
And homozygous dominant for plant height. So homozygous recessive is short, so dominant has to be tall. You with me? Are you okay there?
Yes, ma'am.
Good. Okay, so the genotype of a white flowering short plant. Okay, so now it's asking for genotype. So you're giving me letters. Okay, so what is the genotype of a white flowering short plant? It's going to be?
I want
Big-
to say small f, small h, ma'am.
Okay, but you need to tell me the full genotype. So it's going to be small f, small h, small h. Okay?
Sorry, ma'am. I forgot about that.
Yes, so that's how you'll get your 2 marks. Okay. And then the 3.3.3, something that you do have to learn off, unfortunately, state Mendel's law of dominance for 3 marks. They love to ask this. So please do learn this. Learn the law of dominance, the law of segregation. There's one more, I can't quite remember now. But yeah, you need to learn Mendel's laws. So what is Mendel's law of dominance? Anybody want to tell me a small part of it and everybody can add to that?
It is when 2 individuals with contrasting pure breeding characteristics are crossed. The individual of the first generation will all resemble the parent with the dominant characteristics.
Correct. Very good. That is perfect. 100%. You get 3 out of 3. So you get a mark for when 2 organisms that are pure breeding, that's a mark there, which have contrasting traits. That's another mark. Are crossed, then all the individuals from the F1 generation will display the dominant trait. That will be a 3rd mark. There's another way of saying it. You can say if an organism is heterozygous, you get a mark for heterozygous. The dominant allele, another mark, will determine the phenotype of that individual. That's another mark. Okay, so you can explain it that way as well, but the answer that our young lady gave now is perfect. Okay, so please make sure that you learn that. Don't lose marks in something as simple as that, as giving a definition. Okay. They also like to throw this in the give the biological term. Maybe they can give you the definition and ask you for the biological term that goes with that definition. So you do need to know Mendel's laws. Okay, let's move on. Okay, I might have done this question. Sorry, this is the wrong question. That's actually a DNA question. Excuse me, sorry. Okay, so human blood groups are controlled by multiple alleles. How many alleles control blood groups?
3 alleles.
How many? Sorry, 3.
Yes.
Okay, you can also just say many. You'll still get a mark. 3 or many. Okay, actually be specific, say 3. Yeah. Which 2 alleles are co-dominant in the inheritance of blood groups?
A and B, ma'am.
Correct. So you must write IA and IB. Okay, so don't just write A and B. You must say big I with the A in superscript and big I with a B in superscript. Okay, you with me?
Yes, ma'am.
Okay, let me just make this a bit bigger. There you go. Okay. A man is heterozygous for blood group A and marries a woman who has blood group O. Use a genetic cross to show the phenotypic ratio of their offspring. Okay, so this is for 7 marks. You're always going to get a cross to do. Okay, so how is this going to be done? What is the first line that you're going to write? What's the first line you're going to write?
P1.
The phenotype.
Your
Correct.
phenotype.
Okay, so on the left, you're going to have P1. Do people need me to type this out? Sorry, I don't know what this is. Okay, I'm just making this small and putting it here so that I can use the space and type this out for you. Okay. The first line is going to be P1. It'll be in not superscript, subscript. Okay. And then you're going to write phenotype. And then you're going to say the phenotype of the parents. So it's going to be
Blood group A and blood group X, blood group O.
crossed with blood group. Oh, I'm just writing it this way just for space. Okay. Blood group A and blood group O. Okay. Now what's their genotype?
It would be capital I and subscript A.
IA. Oh goodness.
It's 2 recessive I's.
Yeah, let me just make this bigger so that I can see what I'm doing. Can you still see my screen? I hope
Yes,
so.
ma'am. We can.
Okay, good. Okay. Oh, my superscript button is gone. Okay, I'm just gonna write it like this. Okay, you're gonna understand how it's supposed to look. Okay, it's supposed to be a big I and A in superscript. Okay, so it's gonna be I, big A, small I, crossed with what?
2 recessive I's.
Small I, small I.
Yes.
Okay. Okay. Then what do we have here? What must we write in the sum?
Meiosis.
Meiosis. Okay. Then we write the gametes,
Gametes.
which are
An I with a
IA.
superscript A and then
IA.
a
A small
recessive
I.
I.
And then 2 small
Yes.
I's.
Another small I and then another small I.
Yes.
Correct. I don't know why this is changing automatically.
Ma'am, it's because of the commas. It's a grammar correction.
Oh, God. Just putting spaces. Oh, no. It's too smart for me. Okay. There you go. Next. Now you're going to do your, then it's fertilization. Okay, then you're going to do your Punnett square. Okay. Does everybody know how to do that?
Yes.
Yes, ma'am.
I don't need to draw a table.
No, you
No,
don't.
ma'am.
No,
You sure?
ma'am.
If anybody wants
ma'am.
me to draw a table.
Positive.
Okay, good. At this stage of Matric, you should know how to draw a Punnett square. Okay. Okay, so you got your Punnett square there, where each gamete is going to be crossed with every other gamete. Okay, so you'll have your IA crossed with your small I, and crossed with your small I. Okay, so each gamete is crossed with every other gamete. Okay, that's what occurs in your Punnett square. Okay, then you're going to say F1 generation, your genotype. What's the genotype going to be?
One
Just do that quickly.
big I A
Big I A,
and then a small I.
small. I need to put a comma here. Small. Yeah.
Then there is another big I with an A in superscript and another small I, ma'am.
Sorry, what did you say?
A big I with a superscript A, ma'am, and then a
Yeah.
small I.
Oh, I wrote this wrong. Sorry. That is correct. That is correct. Next. Oh, for goodness sake. Okay. Does everybody understand it if it goes all wonky like this?
Yes, ma'am, I understand it.
Yes, ma'am, it's fine. Don't worry.
Okay.
And
Because it's
two
driving me
small
nuts.
I's and two small I's, ma'am.
Okay.
Yes.
Great. Fantastic. Okay, now the phenotype. Okay, what's your phenotype looking like? Two individuals with
Group A and 2 with group O.
group. Oh, okay. Okay, so this is an example of where they can use it's not your typical Punnett square type of question. Okay, so sometimes they use blood groups for mostly like paternity type questions. Okay, or like crime related type of questions, but you can also get blood groups in a normal Punnett square type of question. Okay. Okay. Yeah, okay. So now 3.3. Hemophilia is a genetic disorder caused by a recessive, excuse me, a recessive allele on the X chromosome. It is a sex-linked condition. A hemophiliac female marries a normal male. explain why all her sons will be hemophiliacs for 4 marks.
Ma'am, I would say because the sons get the X chromosomes from their mother and the Ys from their father.
Yes, that's a very good answer. That is correct.
I would say because the mother has a recessive ally, which could also be given off to her offsprings. Therefore, one of her sons could also get hemophilia.
Okay, so you could say that, but you probably get 2 out of 4 for that type of answer because you need to be more specific. So you need to say that the mother has 2 recessive alleles. Because she is a hemophiliac and hemophilia is a recessive condition, she has 2 recessive alleles. So any allele that she contributes to her children is going to be recessive. Okay, and then you have to say that the males only need 1 copy of the gene for hemophilia to be hemophiliacs because they have 1 X chromosome, which comes from the mother. Okay, because the father contributes the Y chromosome. The mother obviously cannot contribute that Y chromosome because she doesn't have it. Okay, so the X chromosome that the mother contributes will have the a hemophiliac gene on it because she is homozygous recessive, which means both of the chromosomes, both of the X's that she can contribute, both of them have that same recessive allele, and males only need 1 copy of it to manifest the condition. So therefore, all her sons will be hemophiliacs because they have a single copy of this hemophiliac allele or gene. Okay, does everybody understand that? That is the type of full answer we're looking for. And you need to be super specific and sort of explain how this is going to occur. Do you understand?
Yes, ma'am.
Did everybody get that?
Yes, ma'am.
Yes, ma'am.
Okay, if you need me to repeat or anything, or if you need us to put it in the chat or something, please just feel free to let us know, okay? Okay, let's move on. Okay, Tay-Sachs is a disease caused by an autosomal recessive allele. Children with Tay-Sachs disease lose motor skills and mental functions. Over time, the children become blind, deaf, mentally retarded, and paralyzed. Tay-Sachs children die by the age of 5. The pedigree diagram below shows the inheritance of Tay-Sachs in a family. Give Charlie's phenotype. Okay, so this is a pedigree diagram. They've given you names of the children and the parents. Okay, so what is Charlie's phenotype?
Charlie is the squares are males, right, ma'am?
Yes,
Is a male with Tay-Sachs.
correct, because it's colored in.
Yes, ma'am. And it's also a square.
Yeah. Okay, so it's male with Tay-Sachs. And Portia's phenotype? Genotype,
Female.
sorry, genotype.
She's a female carrier of Tay-Sachs.
Correct. Oh, but you need to say the genotype. So if she's a carrier, what's her genotype going to be? They've given you the letter to use.
First X with a N on the top, an N on both X's, sorry.
Okay, so this did not say that it is a sex linked. It says autosomal. Okay, so that means it's not sex linked. So you should not put it on an X chromosome. Okay, it's an autosomal disease. So it's going to be just your normal genotypes and phenotypes. Your normal genotypes.
Oh, sorry, ma'am. I understand. I understand.
So just watch out for that. Okay, so what's a genotype?
Two recessive ends.
If it was two recessive ends, then Portia would have Tay-Sachs, and Portia does not have
Sorry.
Tay-Sachs.
Sorry, two
Ma'am.
dominant ends.
No, Portia is a carrier because
Big N, small n, ma'am.
So that means she'll be heterozygous, big and small.
yes, correct. She's a carrier, and how do we know she's a carrier?
She has a daughter with Tay-Sachs.
Correct. And the daughter would have had to get a chromosome from each parent because it's a recessive condition. Are you with me?
Yes.
Yes, ma'am.
Okay. What is Bill's genotype? Obviously, it has to be a carrier because Charlie has Tay-Sachs. So his genotype is
Big N, small n,
correct. Why?
because he carries the recessive gene.
How do we know he's carrying it?
Because one of his children has the disease.
But he himself does not have it, okay? Because it's a white square.
Yeah.
Okay, is everybody understanding?
Yes, ma'am.
Good. Okay, explain why Patrick is normal, but a carrier of Tay-Sachs. Patrick is here. Okay, so he is the husband of Portia. Explain why he is normal, but a carrier of Tay-Sachs. How do we know he's a carrier?
One of his kids
Because
has it.
oh, sorry, go.
One of his kids has Tay-Sachs, so he has to have at least one recessive.
Correct. Okay, so Patrick has to contribute one allele. Okay, and since Elizabeth has Tay-Sachs, remember she's a female. Would need 2 copies of this, one from each parent because it's a recessive condition. So Portia has to contribute one. Patrick has to contribute one in order for Elizabeth to present Tay-Sachs disease. Okay, so since Elizabeth has Tay-Sachs, she had to inherit one recessive allele from each of her parents. Is everybody with me?
Yes.
Yes, ma'am.
Okay.
Yes,
Yes,
ma'am.
ma'am.
Okay, fantastic. Okay, so you're finding these questions manageable so far?
Yes, ma'am.
Yes, we are.
Okay, good.
Yes, ma'am.
These are from past year papers, so it's good that you are able to do them. Okay. Scientists wanted to determine which type of inheritance accounted for most of the selected genetic disorders in dogs. To do this, they sequenced the genomes of a large number of dogs of the same breed that suffered from genetic disorders. The results of the investigation are shown below. A pie chart showing the type of inheritance for genetic disorders in dogs. Okay, so 10% is sex-linked, 8% is other, 11% is autosomal dominant, and 71% is autosomal recessive. Okay, so 2.1.1. If 2,000 dogs were studied in this investigation, how many dogs had disorders that were caused by autosomal dominant inheritance? Show all calculations. How are you going to do this? It's going to be 11% times 2,000 dogs. Is everybody with me on that? Fairly easy.
Yes, ma'am.
Is everybody
Yes,
with me?
ma'am. Yes, ma'am.
Okay,
Yes,
good.
ma'am.
Okay. Okay, so it'll be 11 over 100 times 2,000 equals?
220.
Correct. Okay. State 2 ways in which the scientists could improve the reliability of their results. Oh, they love to ask this question, guys. Please learn this. Learn this off. How do you increase the reliability? You'll definitely get this question in some section in your paper. It's not only in genetics.
They increased sample size and repeated the experiment.
Correct. Okay. Okay, so if you repeat the investigation or you take a larger sample size, you will improve the reliability. Okay. State one factor that was kept constant in this investigation. What factor was kept constant? A clue is to look for the word same.
The breed of the dog, ma'am.
Okay, correct. That is 100% true. The breed of the dogs were kept constant. Okay. That applies to other questions as well. If you see they say the same this, the same that, the same amount of time, the same breed, the same amount, all of that. Okay, those are things that were it was variables that were controlled or kept constant. Okay. Next question, sorry. Explain why there is no need to keep the age of the dog constant in this type of investigation. Why do you think they did not control for age? Any guesses?
Not from me, ma'am.
Could you explain this one, ma'am? I'm not sure either.
Okay, so explain why there is no need to keep, so they're telling you there's no need to keep the age of the dogs constant in this type of investigation. So what are they investigating? They're investigating the type of inheritance of genetic disorders in dogs. So would this be affected by age?
No.
It would not be affected because they are either born with this trait or not. Do you understand? So age does not matter because the disorders are inherited. So they will be born with or without it. It's a presence and absence thing. So it's not affected by age. Okay, because it clearly says that they where's this? I'm sure I saw genome somewhere. No, but to do this, they sequenced the genomes of large numbers of dogs. Okay, so they did not base it on outward characteristics. They actually sequenced the genomes of the dogs. So they could clearly tell, okay, this dog does have the gene for something. this dog does not have the gene for something. Okay, so age does not matter because the genes for the particular diseases are either there or not. Is everybody following?
Yes, ma'am.
Do you
Yes, ma'am.
understand? Okay,
Yes, ma'am.
so this is the out of the box type of question. So keep it in mind for future questions. Okay. Provide a conclusion for this investigation. They also love to ask this question, guys. So you must be able to write a conclusion. What type of conclusion would you give for this? You've got the results there? It's only for 2 marks, so just give it a try.
Autosomal recessive disorders are most common in dogs.
Very good. So, sorry, autosomal recessive genetic disorders are the most common form of disorders found in dogs. That would be an adequate conclusion to draw. Okay, so it's, yeah, I would go that one too. Can't think of anything else right now.
Yes, ma'am.
Okay, did everybody get that? So just look, it's just looking at your pie chart and taking a conclusion from there. Okay. If this question was for more marks, you might have to elaborate a little. Okay. Because they're followed by autosomal dominance and then sex-linked, and the least was other types of genetic disorders. Okay.
Yes, ma'am.
Okay. Another pedigree diagram. Okay. The pedigree diagram below shows the pattern of inheritance of a certain genetic disorder controlled by a recessive allele. The dominant allele is represented by big N and the recessive allele by small n. Father and mother are there, and two of their children have this disorder. They are the affected male and female. Explain why both parents must be heterozygous for this characteristic. Very easy question, guys.
Ma'am, could you say it's because they have a daughter with the disorder? So therefore they'd have to have both donated both or some sort of X or one of the recessive gene to get the daughter with 2.
Okay, so this is not sex-linked because it's telling you dominant and recessive allele. Okay, it doesn't say anything about sex-linked here. So don't just assume that it is sex-linked. Could just be a normal inheritance type of inheritance. Okay, so rather just leave that out and say because the children are affected and this is a recessive disorder. Okay, both parents have to contribute a recessive allele in order for the children to express it. Okay, so because these children are affected, each parent must have contributed one recessive allele. Therefore, the parents are heterozygous because they do not manifest the disease themselves. Okay, so that means they do have a dominant allele, and then they have a recessive allele because that showed up in the children. Okay. Are you understanding?
Yes, ma'am. Thank you.
Okay. Give the possible genotype for the normal children.
Would it be big N and small n or two big N's?
Yes, so the possible genotype / genotypes. Okay, so you'd say both. You'd say big N, small n, and big N, big N. Okay, so you write both of them. Okay. Provide evidence from the pedigree diagram to show that this characteristic is not sex-linked. What evidence do you have?
Both genders can carry the disease.
Okay, that is true, but that doesn't necessarily mean that it's a sex-linked.
Ma'am, is it because it is controlled by a dominant allele? It doesn't specify that it's sex-linked.
Yes, but it says from the pedigree diagram. So you need to show something that's in the pedigree diagram. We know it from the little blurb on top, but we need to show it in the recessive, in the pedigree diagram now.
Is it
What
because
evidence do we have?
Is it because the male can be a carrier?
Correct, you're on the right track there. You are on the right track, but you're just missing one part of that sentence. So if it was sex linked, what do we know? The male, if the male has a recessive allele, it will show. Correct?
Yes, ma'am.
So what do we know about the father? He is a carrier, isn't that true? Because his children, the two of his children have the disease. Correct?
Yes, ma'am.
The father is a carrier.
Yes, he is,
We've
ma'am.
established that. Okay. So if the father was a carrier, why is he not affected? Because it is not sex linked. If it was sex linked and he has the recessive allele, it should show. Because he would only need one copy of that recessive allele to manifest the disorder. But since it is not sex linked, he has both the dominant and the recessive allele. Are you with me?
Yes, ma'am.
Must I repeat that?
Please, ma'am.
Please repeat, ma'am.
Okay, so the father would have, if I just give you a short, concise answer for 2 marks, the father would have been affected if it was sex linked, because we've already established that he is heterozygous. So you can start off by saying the father is heterozygous. If it was a sex linked condition, the father would be affected, you could say. Because they're just saying affected and normal. So the father would be affected if it was sex linked. Full stop. Since he is not affected, but still possesses the recessive allele, this disease is therefore not sex linked. I hope you're writing that down. Hello?
Yes,
Yes,
we are.
ma'am. I'm copying it down.
Okay. Okay. Okay, so everybody's fine with that.
Yes, ma'am.
Right, let's move on. Okay, blood groups. More questions on blood groups. Okay. So Mr. and Mrs. Fonella are concerned that their baby girl does not appear to resemble either of them. They suspect that the baby they were given at the hospital was not theirs. Mr. Fonella is blood type AB. Mrs. Fonella is blood type B. and the baby they were given is blood type O. Give the possible genotypes of Mrs. Fonella.
Ma'am, is it big IB, small IA, and then big IB and big IB?
For Mrs. Fonella, what's her blood type? Her blood type is big IA, big I
Isn't that
B.
Mr. Fonella? That's
Oh, sorry,
AB, ma'am.
sorry. Yes, sorry. Mrs. Fonella is blood type B. Oh, so it's it's big IB, big IB, or big IB, small I. Andile, you're drag on my screen. Are you busy experimenting with the annotations? I will erase it. There you go. Okay. Okay. So did you get that? So Mrs. Fonella is a big IB, or big IB, small I. Okay, so just the dominant and the recessive forms of blood type B. Okay, the baby girl, what would her genotype be? It'll be
Two
small I, small I.
Nice.
Yeah, two
Okay.
small eyes, ma'am.
Yes. Okay, explain why the baby girl with blood type O cannot be Mr. and Mrs. Fonella's daughter.
Ma'am, there would need to be two
Would this be
recessive
because
eyes.
both parents would have to have blood type O in their genes?
Sorry, what? Can you repeat that?
Would this be because both parents need to have blood type O in their genes and Mr. Ponela does not?
No, okay, so the way you are phrasing that is incorrect. Let me tell you why. You can't say that they have blood type O in their genes. You need to talk about the alleles. So you need to say if the pair, so you need to first deal with the baby girl's genotype. Her genotype is small I, small I. So one parent has to contribute one small I, and the other parent has to contribute the other small I. Now, the mother can contribute the small I if she is IB small I. So she can potentially contribute that, okay? But can Mr. Fonella contribute that? No, he cannot, because he is I big A and I big B. There's no small I in his genotype. So therefore, the daughter cannot be Mr. and Mrs. Fonella's daughter, because she needs a small I from both parents. Is everybody with me?
Yes, ma'am.
Okay,
Yes.
that's how I want you to phrase that. So let me read it to you. The baby inherited one allele for type O blood. So she inherited one allele from each parent. Since her genotype is small I, she has to inherit a small I from the mother and a small I from the father. Mr. Fonella cannot contribute that small I because he is blood type AB. Okay.
Yes, ma'am.
Right. Okay. Explain why the use of blood type for paternity testing is not conclusive.
Because a child can come out with blood type O, which could be different from the parents.
No, not really. Try again. While I cough my lungs out, try again. Think about why it's not good for paternity testing. Why it's not good for conclusivity. So why is it not good for saying definitely, definitely these are the parents? Because think about it, there's lots of people with the same blood type. So if the mother is having an affair with a man, that man could potentially be the same blood type as her husband. Do you understand? So paternity, blood typing can only be used to exclude a particular man. Like in this example here, we can definitely say that Mr. Fonella is not the father. We cannot tell who is, because there may be any number of men that have the blood type that could potentially contribute that small I. Do you understand?
Yes.
Yes,
So
ma'am.
blood type can only be used to definitely exclude a particular man as a parent, but it cannot confirm that a particular man is the father, because there could be many men with the same blood type. Okay. Are you with me there?
Yes, ma'am.
Okay, I hope you're writing these things down, guys. But we are recording the session, and the recording will be uploaded if you couldn't catch anything. Okay. So, but please just ask me to repeat if you need me to. Okay. Using your knowledge of sex chromosomes, explain why the sex of a child is determined by the male gamete.
If the male does not donate the Y, then it will have 2 X's, ma'am.
Correct. Okay. So the female always will contribute your X chromosome. Okay, because the female only has an X chromosome to contribute. Okay. So the normal female has 2 X chromosomes. The normal male has 1 X and 1 Y. The female chromosome, the female always provides that X chromosome. And if an egg cell is fertilized by by an X-bearing sperm cell, then it will be a girl. And if that egg is fertilized by a sperm that has a Y in its genome, then it's going to be a boy. Okay, so the father is the one that contributes the X or Y. Okay, so this is quite a loaded question for 5 marks. So you basically just need to say that the female has 2 X chromosomes, the male has 1 X, 1 Y. That's a mark each. And you can say the female always provides the X chromosome. And if the sperm has an X, then it will be a girl child. And if the sperm has a Y, it will be a boy child. That's where your 5 marks are coming from. In fact, the model answer also shows a Punnett square. Okay, where you could draw a Punnett square to show the female gamete, which is the X, and then the male gamete, which is X or Y, and then you just do your Punnett square where it's XX and the offspring are XX or XY. Okay. And then you just do your genotype and phenotype. So they were lenient in this type of question, but I think it's way too many marks for this type of question, but yeah. So you can either explain it or just show it in a Punnett diagram. So just remember this question is a little bit unusual. Okay, is everybody following so far?
Yes, ma'am.
Okay. Students, is it fine if we continue with our questions? I've got quite a few more.
Yes,
do?
ma'am, that's fine.
Okay, good. We can go until past. Okay, so a mother has blood group B and a father blood group O. They have 3 children and an adopted child. The blood groups of the children are represented in the table below. Which child is adopted? The mother is blood group B, the father is blood group O.
Ma'am, is it Cindy?
Why would you say Cindy?
Because the father is blood group O, so he'd have two small i's, and the mother you said was blood group B, so there wouldn't be a way to get the A blood group. The A required to make the blood group AB the A allele, it would just be the big I with a B and a small I.
Correct. Okay, there's no big IA. Okay, everybody understand that? Oh, is there a question? Okay, no. Okay. Oop. Oh. Right. Okay. That was correct. So we're moving on to the next one. Okay. The table below shows the percentage distribution of blood groups in a province in South Africa. How many genes control the blood groups shown above?
I think it's 1.
How many genes control the blood group? Correct. 1 gene, but many alleles. Very good. Explain how it is genetically possible to have 4 blood groups in a population. How is it possible to have 4 blood groups in a population?
The A blood group and the B blood group carry a recessive I. If crossed, can create a O offspring.
Correct. Okay, so you can say blood groups are controlled by your 3 alleles. You can get a mark for that. And then now in combination, they make up 4 different phenotypic, phenotypic blood groups, where the recessive alleles, the homozygous recessive condition is your blood group O. Okay, that'll give you a second mark. And then you need to do a pie chart using the information in the table above. Okay, so everybody needs to learn how to do a pie chart, because I've seen this in many past papers. So you must know how to draw your graphs. You need to know how to draw your bar graph and your pie chart. It's come up in previous years very often. Okay, so make sure you know how to do pie charts and normal bar graphs as well. Okay, can I move on? Students?
Yes, ma'am.
Yes,
Okay.
ma'am.
Right. Oh, this is the answer. Okay, this will shows you exactly how to put it into a pie chart. So to calculate it, you'd say your percentage times 360 degrees, and that will give you the degrees. Okay, so you use your what do you call that thing? The word escapes me.
Protractor.
Yes, protractor. I'm like set square. No, you use your protractor to get the degrees, and that's how you draw your pie chart. Okay. Okay, they also love to ask this cat question. Okay. In cats, 2 genes on different autosomes, so it's telling you it's autosomal, have the following alleles: black coat color, gray coat color, no white patches, white patches. Okay, so it's 2 different genes, dihybrid cross. 2 cats were mated. Their litter of kittens had the phenotypes shown below at a ratio of 9:3:1. You need to learn that whenever you have a ratio of 9:3:1, it is a dihybrid cross with heterozygous parents. Okay. Name the type of cross represented here. It is a
Dihybrid cross, ma'am.
cross. Give a reason. There's 2 different characteristics being considered. Okay. Give the genotype of phenotype D. Gray coat with white patches. What's the genotype of that?
Small b, small t.
I don't know why you're making this mistake, guys. Tell me the full genotype.
2 small b's, 2 small t's.
Correct. Okay. If you just write small b, small t, you will not get the mark. You need to write small b, small t, small t. Okay, that is the full genotype. Is everybody with me?
Yes, ma'am.
Right. The phenotypes of the parents.
Big B, small b.
Phenotype.
Oh,
Phenotype of the parents. Remember, when you have a 9:3:1 ratio, your parents are heterozygous dominant. So if they're heterozygous dominant, it means they're going to show the dominant traits, but they are heterozygous. So it'll be black coat color with no white patches. But their genotype will be big B, small b, big T, small t, because they do have the recessive allele there. But they are just showing outwardly, they're showing the dominant trait. Okay. That's just something you must learn off, guys. Whenever you have a 9:3:1, your parents are heterozygous dominant. Explain why all the kittens have having a black coat with white patches may not have the same genotype. Explain why all the kittens having a black coat with white patches, this one here, phenotype B, black coat with white patches may not have the same genotype. What's black coat with white patches? What's the genotype for this?
2 big B's and 2 small T's.
How do you know that it's 2 big B's?
It could also be big B, small b as well. ma'am.
Exactly. Okay, so they don't have the same genotype because they don't necessarily have the same genotype because it could be heterozygous or homozygous dominant, because black coat color is controlled by a dominant allele. Okay. Then state Mendel's principle of segregation. What's Mendel's law of segregation?
During meiosis, they'll separate into different gametes.
What will separate?
The alleles.
The alleles on homologous chromosomes. separate during meiosis, and then
one allele from each pair will be present in a gamete.
very good, correct, 100%. Except you need to say the pair of alleles on homologous chromosomes separate during meiosis such that one allele from each pair is present in the gamete. Okay.
Yes, ma'am.
Very good. Okay. Okay. In humans, the allele for unattached earlobes is dominant over the allele for attached earlobes. The allele for the ability to roll the tongue is dominant over the allele for inability to roll the tongue. The female that is heterozygous for both unattached earlobe and ability to roll the tongue is married to a man that has attached earlobe and is heterozygous for the ability to roll the tongue. That's a mouthful. How many characteristics of humans are being investigated in the statement above?
2.
Correct. What is the genotype of the female? She's heterozygous for both unattached earlobe and ability to roll the tongue. So what's her genotype?
Big E, small e, big T, small t.
Correct. What is the phenotype of the male?
Small e, big T, small t.
No, phenotype.
Attached earlobe, ability to roll tongue.
Correct. Okay, remember now, don't get confused with genotype and phenotype. Okay. If they ask for genotype, that is the genes that they require. Phenotype is the look of what is represented. Okay. Okay, C, question C, the genotypes, give the genotypes of all the possible gametes of the male. The male is heterozygous, or the male that has attached earlobes, and heterozygous for tongue rolling. So what is his gametes?
I want to say 2 small e's and a big T and a small t.
So what's the gametes?
1 small e, another small e, a big T and a small t.
Oh, you separated each of them. Okay. So now in a dihybrid cross, you always need to have the two characteristics next to each other. So it'll be big. So put them together. It'll be small e, big T. That's one gamete.
Yes, ma'am.
Okay, and small e, small t, that's another gamete. Okay. Okay, are
Yes,
you with
ma'am.
me? Okay.
Yes, ma'am.
Name Mendel's law that allows an allele for unattached earlobes to show up in a phenotype of a heterozygous individual.
Mendel's law of dominance.
Correct. Okay, that is Mendel's law of dominance because unattached earlobes will show up because it is dominant over attached earlobes. So the dominant trait will show up in the heterozygous individual. Okay. Okay, last question. Hunter syndrome is a sex-linked disorder. Okay, it's telling you it's sex-linked that is inherited as a recessive allele. Small t on the X chromosome. The diagram below shows the inheritance of a Hunter syndrome in a family. Okay, give the phenotype of Peter. Phenotype of Peter.
Hunter syndrome affected male.
Okay, so he's an affected male, correct? Sorry, okay. The possible genotype of happy. Possible genotype or genotypes of happy, who is an unaffected female.
Ma'am, could it be X's with 2 big T's in supertext and a X with 1 big T and a X with a small t?
Correct. Okay, so when they ask you something like this, you know it's going to be the heterozygous condition and the homozygous condition. Okay, so since she's unaffected, it will be it'll be big T. So it'll be X big T, X small t. That's her 2 possible genotypes. Okay, oh sorry, homozygous, it'll be X big T, or X big T, X small t. Homozygous or heterozygous conditions. Identify the female member in the diagram above who is a definite heterozygous. Definite heterozygous. So it's between Ethel and Happy and Patricia. Who is the definite heterozygous?
Would it not be Ethel?
Ethel.
Yeah, yes, that's correct. It's Ethel because Lucky is affected. So she obviously had to contribute an X chromosome there. So she has to be heterozygous. Give the percentage of unaffected males in the diagram. So how many unaffected males? We've got 1, 2 unaffected males in the diagram above. So it's going to be 2 out of
2 out of 5, ma'am.
there's 1, 2, 3, 4, 5. Yes, 2 out of 5 males. Okay, so that is 40%. Okay, explain why. Remember, it's asking for the percentage there. Okay, explain why Lucky's condition cannot be used as evidence that John is his father. Explain why Lucky's condition cannot be used as evidence that John is the father.
Because Ethel carries a recessive allele.
Okay, you need to elaborate because it's for 5 marks.
I don't know, ma'am. Can you give us a helping hand?
Yes. Okay. So, explain why Lucky's condition cannot be used as evidence that John is his father. So it's very similar to the blood typing question now. Okay. Since Ethel is contributing, one allele, okay? Any male, oh wait, it's X-linked. Okay, so I'm just trying to put it in nice words. Okay, start off by saying that since Ethel is contributing a recessive allele to Lucky, John is also contributing a recessive allele, but he is contributing the Y chromosome. Okay, so this is on the X chromosome. Do you understand? So Lucky only needs one copy of this allele for him to show the trait. So since the trait is on the X chromosome, it does not matter which male contributes the Y, because he's definitely going to, Lucky's condition is definitely going to be expressed because he got that recessive allele from his mother. She contributed that X. The Y that is contributed by the father, it really doesn't matter whether it's John or Ben or Jack. It does not matter because he got the recessive allele from Ethel. So Lucky's condition, the fact that he has Hunter's syndrome, does not guarantee that John is the father because he is getting the Hunter syndrome allele from Ethel, his mother, because she contributes the X chromosome. And Lucky only needs one X chromosome to express the trait. Is everybody following?
I understand for the most part, ma'am, yes.
Okay, so you want me to repeat? Although I said that from the top of my head, so I can't exactly repeat it exactly how I said it, but I can try. Anybody want me to repeat that?
No, ma'am, you're good.
You got it. So basically that X chromosome that has the recessive allele is coming from Ethel. So John is inconsequential. It doesn't matter what John contributes because the X chromosome that has the allele for Hunter syndrome is coming from Ethel. Therefore, Lucky's condition is not determined by John. It is determined by Ethel. And therefore, it cannot be used as evidence that John is his father, because there's not, the only thing that John is contributing is a Y chromosome. Okay. Is everybody following me there?
Yes, ma'am.
Yes, ma'am.
Okay. Okay, guys. So that brings us to the end of our questions that I had planned for tonight. Does anybody have any last minute questions they want me to go through, or any questions they want me to answer quickly?
No, I'm good.
Same here, ma'am. I should be fine.
Okay. If anything comes up, please do not hesitate. You can even bring it up in the next session as well. It's absolutely fine. I hope this session was helpful to you guys, and thank you for staying on for some extra time, and I hope you guys learned something, and I know you definitely got some practice in, so you are well prepared for your June test. Okay, I will be doing our next session next week, Friday, same time, same link, same Zoom link. Okay, so and I'm going to be doing I think it's reproduction next week. But if you have any suggestions, please feel free to put it in the chat. If you can please all register, just quickly click the link and it'll just ask you for your email address or something, and your name and your email address, just so that we have a register of you guys, and you'll also possibly receive a message the next time we have this Matric Helpline. Okay, so thank you all so much for joining, and have a great evening.
Thank you, ma'am. You too.
Thank you,
Thank
ma'am.
you.
Enjoy your weekend.
Thank you, you too. Bye. Oh, please feel free to click the link on the chat that asks you to do the evaluation, if you don't mind, please.